A biofilm is any group of microorganisms in which cells stick to each other on a surface. These adherent cells are frequently embedded within a self-produced matrix of extracellular polymeric substance. Biofilm formation is a major threat to medical and industrial sector. Bacterial proliferation changes the pH of the surrounding due to the slime released. Therefore, precise measurement of pH changes induced by the proliferation and metabolism would contribute to the detection of biofilm formation. With this basic idea, let us now have an overview of our project. The environmental changes in the pH due to the biofilm growth in the form of H+ ions is sensed by the ion-sensitive field effect transistor. The ISFET sensor yields the output in the range of millivolts, which is then fed to the hardware circuitry to be amplified in the range of volts. The hardware circuit also indicates the battery level, ready or change battery. The amplified voltage is then fed to the Arduino Uno board, which converts the analog signal into a digital one to trigger the buzzer alarm and to display the pH value and graph. Also, a GSM shield is interfaced with the Arduino board to send a message to the desired mobile number. To start with the bacterial culture, 70% ethanol is the most effective killer of any microbes, so the work area is cleaned with ethanol before culturing the bacteria. A spirit lamp is lit and the mouth of the test tube is shown in the flame to kill any invading microorganisms. The tryptone soya broth culture medium, autoclaved at 120 degrees Celsius, is taken in a 10 ml test tube. The pH of the solution is neutral. "50 microliters of Escherichia coli bacteria is taken in a micropipette and poured into the nutrient broth. The inoculum is then labeled and incubated at 37 degrees centigrade. The major components used are an ISFET sensor, which is gateless with only drain and source terminals. It is chosen for extremely high sensitivity and accuracy. The response time is 10 seconds. The reference electrode is the glass electrode. The TI components are operational amplifier OPA4277, micropower voltage regulator LM385 2.5 volts, dual comparator LM393N. The other components are Arduino Uno board for processing and control, a GSM shield for mobile communication, and a buzzer for an alert tone. This is our PCB design. The blue and black wires are the output ends. The blue, yellow and green are connected to the drain, source and gate terminals respectively. The ICs are powered by a 9-volt battery. The output from the sensor is in the range of millivolts and the amplifier OPA4277 is used to give the output in the range of volts. The micropower voltage regulator LM385 is used to give a constant voltage of 2.5 volts as reference. The LM393N dual comparator avoids the need for a dual supply and is used to indicate the battery level. The red LED indicates device ready and the green LED indicates change battery. The output from the circuit is connected to the analog input pin of the Arduino board, and the ground connections are made. The Arduino is interfaced with the GSM shield by connecting the receiver of the GSM with the transmitter of the Arduino board. And the transmitter of the GSM with the receiver of the Arduino board. The buzzer for sound alarm is connected to the digital output pin of the Arduino board, and the entire setup is connected to the computer using the Arduino software. The drain, source and gate terminals from the sensor are connected as shown. And the entire setup is placed inside the incubator to nurture bacteria at constant temperature. The entire closed device is placed inside the incubator and maintained at 37 degrees Celsius. Voltage of standard buffer solutions with pH 4, 7 and 10 are measured and taken as reference for calibration. Using these values, a linear line is established and the equation of a straight line is obtained. This equation is used to convert the obtained voltage values to its corresponding pH. The experimentation starts by placing the sensor inside the inoculum, and the whole setup is kept inside the incubator at 37 degrees Celsius. The tip of the sensor and the glass electrode touches the inoculum surface. The setup is interfaced with the system to collect the data which can be monitored for every 2 hours in a serial monitor. The data is also saved in an Excel file using PXL DAQ software, and the graph is plotted using SimPlot. This is the code along with the data values obtained at an interval of every 2 hours. From the graph, it is clearly seen that the pH value drops and becomes acidic in the 6th hour. Here is the message when the pH value drops below the set threshold 4.5. The picture in the left side shows the turbidity of the inoculum, which indicates the bacterial growth after the 24th hour of culturing. Biofilm formation is becoming ubiquitous these days and is being a severe threat. Hence, our proposed device shall be used for detection of any adverse microbial biofilm contamination or infection.